Hi, I am Helen Thomas. Let's learn the working principle of hematology analyzers. Hematology analyzers can be used for CBC, hemogram, retics, hemoglobin, etc. The electrical impedance or Coulter principle could be used for counting and sizing of the cells. The diluted blood is passed through a small aperture. There are electrodes that are placed on either side of the aperture. As a cell passes through this aperture, it displaces the electrolyte, creating an electrical resistance, which is recorded as the pulse. And each pulse corresponds to the cell count. The amplitude of each pulse is related to the size of the cell. So here, if a platelet passes through this aperture, it creates a pulse of a smaller amplitude, while a WBC will create a pulse of a greater amplitude. There are analyzers that work on optical principles, often called as flow cytometry or laser light scatter. This method could be employed for counting, sizing, the differential and the detection of cells. Here, the cells are passed through a laser beam one at a time in a fluid stream, often called as hydrodynamic focusing, in which the dilute sheath is ejected through two nozzles at a constant pressure and the cell suspension is ejected through the sample ejection nozzle. Hydrodynamic focusing helps in such a way that the cells are passed through the laser beam one at a time. The light is scattered in various directions depending on the size of the cell. We get the forward scatter, and the forward scatter will help in detecting the size. The side scatter will help us to have an idea about the complexity or granularity of the cell. And if the cells are tagged with a fluorescent dye or a chemical dye, then the fluorescence is also detected at this stage. The detectors or photodetectors that are used in the optical system will capture the scatter patterns to identify and classify the cells. There are advanced analyzers which use multiple angles and fluorescence tagging to distinguish cell populations. Hydrodynamic focusing is a fluid system principle used in both impedance and optical analyzers or in analyzers that uses both these principles. It helps in the alignment of cells in a single file for the accurate counting, and it reduces the coincidence errors. So through this area, only one cell will be passing. So as the laser is ejected in this way, only one cell will be in the path of the laser light. So here you get the forward scatter, and through this way you may have the fluorescent scattered or the side scatter detectors. Cyanamide hemoglobin or non-cyanide methods are used for hemoglobin estimation. Here, the blood is mixed with a lysing reagent which will rupture the RBC, resulting in the release of hemoglobin. The hemoglobin is then converted into a stable colored product, and the absorbance is measured spectrophotometrically. The absorbance is directly proportional to the hemoglobin concentration. The ferrous hemoglobin, the Fe2 plus hemoglobin monomer undergoes oxidation by the ferricyanide and forms Fe3 plus hemoglobin, that is the ferric form, the methemoglobin monomer. And then the potassium cyanide will act on this ferric hemoglobin, which will convert it into a cyanamide hemoglobin, which is a stable colored product. And this absorbance of cyanamide hemoglobin is red at 540 nanometers. This is a small animation of what we just discussed. This is the hemoglobin estimation. It is photometrically read. Here, the hydrodynamic focusing the laser system will detect the cell count and the size. Thank you.